Hi, this is Miss Linton. You can sit in my office if you want to. Hi, this is Miss Linton, and this is 7th period support for HBio. Say hi.
Hi.
And then you are going to want to be able to log in with something so you can do some of the practice questions that we didn't have time to do in class. First thing I want to make sure that you understand is the confusion between whether or not proteins or DNA would be our hereditary information. And the concern was there's only how many bases in DNA? 4. And how many different amino acids? 20. So it seemed like logic-wise you could do more when you have choices of 20 than you have with choices of 4. But because DNA there's so much of it, it allows for us to still be able to create unique genes because of the sheer amount of DNA. When you did your hyperdoc, how much DNA do we have? Enough all the way to go to the moon. Good. And we talked about the classic experiments, Hershey and Chase with their bacteriophage. And we talked about labeling the DNA with radioactive isotope. Which one? Phosphorus. You're going to remember that. You don't have to memorize these isotope numbers, but I just want you to remember phosphorus is found in base sugar. What is DNA made out of? A base, sugar, and phosphate. Remember? Base, sugar, phosphate, right? So that's why you can use radioactive phosphorus to label the DNA. Does that make sense to you?
Yes.
Okay. So you don't know this part, but viruses are made out of proteins and they're made out of nucleic acids. And so they labeled the radioactive, labeled the DNA with radioactive phosphorus because then they could trace it. And they were able to find when those viruses infected bacterial cells that radioactive phosphorus was found inside the bacteria. And that gave evidence that it was the DNA that was what they used to invade and take over the host cell machinery with, and that it was a nucleic acid. And we talked about how they used blenders to separate and then a centrifuge. Other classic experiment you need to know are Watson and Crick. And Watson and Crick, because they came up with what of DNA? Structure of DNA. And they used evidence from some other experimenters. One of those was Chargaff. Do you remember what Chargaff did? No? I'm listening. Sorry, I can't hear everything. A equals T, and the number of G's equal C's. And for any one species, the A's and T's and G's and C's are a specific number. That part can vary. But however many adenines you will have the same number of thymines. However many guanines you have the same number of cytosines. Okay? And then another piece of evidence they had was from Rosalind Franklin and also Maurice Wilkins, but she's the one who gave it to them. And they would shine what through crystallized DNA? X-rays. And those formed a pattern. And just like you can tell if a bone is broken or not when you see an X-ray, they could tell structure from the pattern that was made by the X-ray going to the crystallized DNA. And they could see that it was of constant diameter and it was helical in nature. So now they have, okay, it maintains a constant diameter, it's helical in nature, the number of adenines equals thymines and guanines equals cytosines, and they were chemists, they knew about bond angles, and that's how they came up with the twisting double helix. Okay? And then from that, if you iron that helix flat, by the way, where are my 0 period students in here? 0 period students, we are going to finish up we need to discuss in the chapter, and you know how we have time during the group test, how we have more time out at the front than what we need? We are going to, I decided, we're going to take our quiz the same day we take our group test because we have time.
So it's going to be Wednesday then?
No, it'll be Wednesday. Tomorrow, no quiz for you tomorrow. You will do it on Wednesday because they have longer to go over and review than you do. Does that make sense, 0 period?
Yeah.
So you'll take, when you first come in at 7:20, 0 period, you're going to take your quiz on Wednesday, and then we'll go over the quiz, and then you'll take your group part. And then Thursday, you'll take your multiple choice part. Okay? Because we don't have the same amount of time. Yes?
Can we come in early on Wednesday, or will we have enough time no matter what?
Oh, we'll have enough time because we always have, because I give you the same amount of time as I give all my other students. And so you have more time in a 0 period. I only give you, how much do I usually give you for each half? 37 minutes, and we have an hour-long class. Yeah. Okay. All right. So I just wanted you to know that 0. Does that make you feel better?
Yes.
Okay, good. All right. So we know that it is a double helix in structure, and you can review that. That's something that you learned last year as well, that it's made out of 2 chains of nucleotides, and nucleotides consist of a base, sugar, and phosphate. You know that a purine is connected to a pyrimidine. Adenine is considered adenine and guanine are purines. They have 2 rings, and thymine and cytosine are pyrimidines, and they are just 1 ring. We know that it's anti-parallel, and when we say that, we're referring to the sugars. The sugars go 2 different directions. When does that become important? When you're going to replicate or transcribe the DNA, the enzymes work in 1 direction. They read from the 3 to 5, making complementary 5 to 3. And we said leading strand and lagging strand. Good. All right, so here's some questions right here. Why don't you log in? This would be the best way to prepare for your quiz, is doing all the questions. Let's log in as your favorite ice cream. Once I remove all these students. Log in as your favorite ice cream. And I'm going to pause you just for a minute. Okay, so let's see how you did. So in the Hershey and Chase experiments, radioactive phosphorus was found inside the bacterial cells, not outside, because that is what the virus injected in was its nucleic acid, not a protein. According to chart, good job. Everybody who answered got that right. You need me to go over that at all? Don't make weird noises. If in an organism, this one's perfect, do you need me to clarify this at all? Okay. Yay! Okay. And if you're at home and you don't understand this, what you do is just problem solve out of 100%. So if you know one of the 4 numbers, let's say one of your 4 numbers is, let's do this one together. What if one of my 4 numbers is 40%? And let's say it's thymine. Could I solve for all the rest?
Yeah.
Okay, tell me how would I do it? Adenine is 40, and that equals, what do we have left? And that would have to be shared. Yeah? So proud of you. Okay. Now, let's talk about DNA replication. DNA replication is semi-conservative replication. Each old strand serves as a template for a new strand. Okay, so all you have to do is bring in the matchy base. So if I have an A, I bring in a T, a G, C, C, T, and I can have T's because I'm making more DNA. And when would I make more DNA? When? What part of interface? As stage of interface. Exactly. Okay? And these, in fact, are what? Sister chromatids held together by a centromere. They're going to separate during anaphase. One thing to get in your head. When you're doing DNA replication, you're replicating every bit of DNA in that nucleus. When we're doing transcription and translation, we are focusing on a gene. And we are making mRNA copy of just that one gene. And then we're going to translate that one gene into a protein. Now, you could do multiples at once, but what I'm saying is you're not making mRNA copies of everything, just the genes that are under consideration. Okay? And here we talked about how it serves as a template, and these would be your sister chromatids, and we know how to do matching bases. Let's review the enzymes that are required. Okay, do you know what helicase is for? Perfect. What about DNA polymerase? Yeah, bringing in those free nucleotides. And what's ligase? It seals the phosphate sugar backbone. All right. And let's do some questions. And I'm going to pause you just for a minute. Okay, so let's check our answers. Okay, so the parental strand is ATTGGC, would code for the daughter strand, and 21 of you got this right. It's whatever is complementary. So A binds with what? T, T with A, T with A, G with C, G with C, and C with G. And that's what most of you picked. What happened here to these people? What did they do? They picked the exact same as the original. Remember, it's semi-conservative replication. The original strand serves as a template for the new strand. If it's an A here, you're not bringing in another A, because then it would be the exact same thing. Remember, it's complementary base pairing. So based on what's here, if this says left, this one's going to say right. If this one says up, this one's going to say down. If this one says in, this one's going to say out. It's complementary base pairing. So you're not going to say left, left, right, right. You see what I'm saying? It's what matches that in order to store that code. Check with your seatmate and see if they understand that. Go ahead. Shadowland, sunshine. Ask the person next to you. Anything you want to ask me? All right. So number 2, everybody who answered got that right. Whoops. Okay then. Okay then. All right, so DNA polymerase is what brings in the free nucleotides, and DNA ligase seals the phosphate sugar backbone. What does helicase do? One. Okay, so here we go. Give me your DNA. Give me your DNA. DNA unwinds and unzips helicase. This is my leading and this is my lagging. Then I'm going to use DNA polymerase. Boom, boom, boom. When I boom, boom, A, G, C, and then on my lagging strand, boom, boom, boom. Then these are going to ligase. Then these are going to twist up and form sister chromatids held together by a centromere, separate during anaphase. Good. All right. Now, talking about RNA, RNA is different than DNA because RNA has the sugar. RNA has the sugar ribose, whereas DNA has the sugar. So when you bring in a nucleotide, that nucleotide will not contain deoxyribose. Like when we were making DNA, it's going to contain ribose. Is the phosphate part the same? Yes. Are the bases the same? Yes. All except when you want to bring in thymine. Yeah, there are no thymines in RNA. There's thymines in DNA. So what nitrogenous base replaces thymine? Uracil. Good. Okay. There are 3 kinds of RNA. Messenger RNA takes the message from the DNA, and this you can just leave it on top of that other test I have in there. Thank you, sweetheart. You feeling better? Okay. This is when this gets transcribed, when this mRNA gets transcribed, this is the message. This is the gene that we're looking for. Now, when you initially transcribe that mRNA, is that the done deal? Whatever, here's your DNA, you make your mRNA, is that it? What could we do? Well, yeah, we need to do translation, but do we always translate exactly the mRNA transcript that we copied? What could we do to this mRNA? Cut out the introns. What's an intron for one protein product may not be an intron for another protein product. This is how we can use the same sequences in the DNA to ultimately get different protein products depending on how we cut and splice. Yes?
What are introns again?
Introns are the part of the transcript that we cut out, which is annoying, I know, because they're called introns. But the reason why the ones we keep are called exons is because that's the DNA that is expressed. Exons are expressed. Say it. Exons are expressed. Exons are expressed. Okay? And it's the introns that we cut out. Okay? You also transcribe transfer RNA. Transfer RNA has a job to transfer something. What will it transfer? Amino acid to where? The ribosome, the workbench. It'll know where to land based on the mRNA. Because the anticodon is located on the tRNA, which you can see right here in this image. So if this anticodon right here is CGC, then the mRNA it would land on would have to be what? GCG. And if this is the mRNA, what was the DNA that coded for it? C. Do you get that? Right? Okay, so the DNA is transcribed into the mRNA, then the mRNA is translated or matches then that tRNA. Now, what do I look up to find out what amino acid it's going to carry? The mRNA. How do you remember that? The mRNA is the message. So I could go and look what that would carry. I'm going to go to this codon chart. It's GCG. It's the message. So I'm going to go look up GCG. So I'm going down here to G. So I know I'm down here somewhere in this range. GC. So it's now here. One of these four. And GCC. It's this one. What is it? Alanine. Did it even matter what the third nucleotide was? No, I was still getting alanine. And so if I have a point mutation on the last one, it wouldn't make any difference, right? Instead of GCC, if I had accidentally done GCA, I'd still get alanine. But what if instead of GCC, I did GUC? Would that have made a difference? Yes. Okay. And in that case, I'm now coding for valine. And that could be bad because that could affect how that protein folds, right? And so as a result, maybe that protein would be non-functional. Okay, and the third kind of RNA that we are looking at right here is ribosomal RNA. And the ribosome itself, the workbench, is made out of rRNA and proteins. Can you see that there's two subunits in the ribosome? And the mRNA first hooks up with the small ribosomal subunit, and then it hooks up with the large. And then they're going to, when it hooks up with the small, that's when the first tRNA lands, and then it kind of locks the cap on the large ribosomal, and then you just start going. And you're going to continue to hook those amino acids together that the transfer RNA brought until you reach a stop codon. Good. All right, so when we look at this picture, transcription is when you use the DNA to make the mRNA. Now they call it mature mRNA. What must have happened to it to make it mature? It must be wearing a cap and a tail. And the cap is 7-methylguanosine, and the tail is 100 to 200 adenines. And you must have cut out the introns before it leaves the nucleus. Perfect. Okay? And then translation, when that mRNA is out here, we can see every 3 bases on it is called a codon, which is complementary to the anticodon. Land. You keep hooking the amino acids together until one of those codons says stop. Yes?
Why do we cut out the introns before it leaves the nucleus? Like, why can't it leave with the introns?
If the introns are in, it's not coding for the right protein that we want, because our DNA can code, one sequence of bases on the DNA can code for several different proteins just depending on how you cut and splice it.
Oh, so you're just cutting to find the right protein?
Yeah. Yeah. It's very specific to each protein. Yeah. Okay, so this is the central dogma, okay? So DNA replication, that's the first part of our central dogma, DNA to DNA. And that's going to happen when we get ready for what? When is this going to happen? No, that's not when it's going to happen. Yeah, we're going to look, it's going to happen during the S stage of interphase. And it's when we're getting ready to do what? Mitosis, when we want to make more cells. When cells just want to function. This is making a copy of the code. This is using the code. It's how I use the code when I do transcription and translation. How do I use it? I make mRNA. I use that mRNA to build a protein. This is how my DNA codes for proteins. This is just making sure I get the code right in all my daughter cells. All right, check with your bio buddies. Shadowlands or sunshine? right? This is showing you making the mRNA. This is showing the processing, cutting out the introns and the cap and the tail. This is showing you, in this case, if this was exon 1, exon 2, exon 3, then this is exon 1, exon 2, exon 3. But if I considered this to be an intron 2, then this would be gone, and this would be shorter, right? It would just be exon 1 and exon 3. And this would code for protein, let's say protein number 2, whereas this sequence codes for protein number 1. So I can get different proteins from the same original sequence of DNA. All right? Okay. And we did the codon chart. Oh, stop. Oh, wait. My bad. My bad. Here we go. Okay. It's only one question, so answer fast because I'm not going to pause it. That's so easy. That's an easy question. Lemon squeezy. Apparently not. Okay, let's read this question thoughtfully. Which RNA carries the amino acids to the ribosome? Transfer RNA. One of you picked mRNA. Does the mRNA carry any amino acids? No. Does it even touch an amino acid? No, it does not. It carries the code from the DNA out into the rest of the cell. rRNA is the rRNA is what is the ribosome, along with some proteins. Okay, so tRNA transfer. That is surprising to me that you missed it. Turn towards your bio buddy. Ask your bio buddy, Shadowland, sunshine, is it cleared up? Was it a silly mistake?
Sunshine on a cloudy day.
All right, so this is just a review. Here's the ribosome in the background. tRNA1 lands, tRNA2, and what's going to happen is this chain always gets dumped on the new guy, then he's going to leave. We talked about the 3 different sites on the ribosome. There was the A site, do you remember? P site, and the E site for exit. Environmental Protection Agency, EPA. Okay, so the new guy always lands in the A site, and the A, when I talk about sites, these are on the ribosome. And it's actually a ribozyme. It acts like an enzyme. And as the ribosome shifts, whatever tRNA was in the A site is going to end up being, let's see if I can grab this. No, I cannot grab it. There. Then he's going to end up in the P site when this is on, and this guy will end up in the exit site. So the ribosome moves along, catalyzing the reaction to put those amino acids together. All right, here you go. Pause you just for a minute. All right, okay. So then this, you can look in your book to kind of review the process. And like I said, there was a really good diagram here. I like how it puts it all together. You could literally Google transcription and translation and probably watch 100 videos on it, you know, to review that process. Maybe this one will be better. I do not know. It is a mystery. Charlie Brown. Is it a good question? Yeah. Okay.
We need the chart.
Oh, okay. Here's your chart. Do you know how to do it? No. Okay, so here, let's double it up. Okay, give me the DNA sequence. Wait, go slow and loud. Go.
T A C G T T C C A C T.
Okay, good job, Winnie. Okay, did I get it right? Okay, so now let's make the mRNA. Tell me what would be complementary here. What? A-U. Because there are no T's because we are in what? RNA. A-U-G. I'm going to just circle that. That's one codon right there. What's the next one? There's another codon. What's the next one? What's the next one? Perfect. Okay, so if I ask you what the amino acid is, we don't need to figure out the tRNA here, We don't need to figure out what the anticodon is, but you could, right? All we're concerned about is what would be the amino acid that we got from each of those, right? Because I could say what the tRNA would be. It would be UAC, right? GAA, or GUU. Sorry, my bad. GUU, and then CCA and ACU, right? But I don't care about that because all I have to do is look up the mRNA. So AUG, what does that get me? AUG, meth. You're not doing that. Methionine, it's always the first amino acid. What's the next one going to get me? CAA. CAA gets you what? Glutamine. Okay, the next amino acid, GCU. GCU, doesn't matter. Alanine. Okay, and what does UGA get me? UGA is what? Stop, there isn't one. I'm done. So how many amino acids did it get me in a row? 3. Ask your bio buddy if you get that. Shadowland, sunshine.
In the middle.
Yes. Wait, I can't hear. Shh, go.
When it says the sequence of amino acids, does that automatically mean you should be thinking RNA too?
Well, the only way to get amino acids is if you have a tRNA. And the only way to know the right tRNA, you have to have the mRNA. But when I look it up, I really only have to pay attention to the mRNA. I mean, I could figure out the tRNA here and what would be complementary, A with U, U with A, G with C, and I could say this tRNA would carry that. But all I really needed was the mRNA in order to figure out what it was going to carry. Yes? Wait, guys, don't talk over the top because I need to be able to hear. Go.
Are we going to be, do we need to worry about the codon chart for the...
You don't have to memorize it, but you should know how to use it. Yes?
The DNA is copied into the mRNA, and then the tRNA is copied from the mRNA?
Yeah, well, it's not that you use the mRNA to copy the tRNA, but the tRNA is made, all RNA is made from DNA. So when we look at our DNA, there could be sections along here, okay, that code for mRNA. There could be sections along here that code for tRNAs. And there could be sections along here that code for rRNA, right? All RNA comes ultimately from DNA. But once you have your mRNA, the tRNAs will land according to what's on the mRNA.
I guess I'm just confused, like, why, if it says, like, this is the DNA strand, what would the amino acids be? Like, why is it the reciprocal?
You don't have to think about it as the reciprocal, okay? What you want to keep in your mind is that the DNA serves as a template to form the mRNA. Okay, so now I have this mRNA. This is what I look up every 3 bases in order to know what my amino acids are.
Do we even need to change it to the mRNA?
Yes, yes, you do.
The chart is based on the mRNA.
Yes. Guys, don't talk over the top in case other people have questions. Go. Shh.
Wouldn't the last one be like lysine or whatever because it's actually GGU instead of GCU?
I'm looking up UGA.
Yeah, but like the second to last one. That
Oh,
one?
GGU. Did I look up the wrong one?
Yeah.
GGU. Yes, it should be. What did I do? What did I look up? Did I look up CCA?
No, you looked at GCU. 'Cause it was
Oh, I thought that was a C. Oh, okay, I'm all, did I look at the wrong? Yes, good catch. Okay, last but not least, my preciouses, is to review your mutations, right? That was the very end. Okay, and we talked about the causes of mutations, so you want to review that. We talked about transposons and jumping genes, where DNA moves around. And we talked about a really bad mutation, which was frame shift mutations, because when you delete a base, right, you're throwing off all your series of codons. It's like really important that you copy that exactly right. And then we talked about point mutations, that some mutations cause no problem. If you end up getting the same amino acid, it's a silent mutation and you're not worried about it. But it could be where it all of a sudden codes for a stop codon right in the middle of your sentence, and then it doesn't work, right? Or it puts in hot jalapeno peppers jumping around singing a song with raisins. It puts in something that doesn't make any sense, right, to what you're trying to build. So some mutations are worse than others. And that is it. Smarty McSmarties. Use the questions at the back of the chapter to help you too. Okay? Yes? Yes, I can. In just 1 second. Okay, and make good choices. Don't stay up too late. Have a piece of toast. Yeah.